If you are an aspirant of CSIR JRF with life sciences, if you are willing to learn all the concepts of cell cycle regulation in just one go, and if you are tired of learning the names of factors and proteins involved in cell cycle, stay tuned, this video is for all of you. Hello everyone, I am Khalid, welcome to my YouTube channel BioBlogger, and today we are going to talk about the cell cycle regulation. While I was preparing for this exam, I realized that most of us leave this topic untouched because of two main reasons. Firstly, when we don't get to know how important this topic is from examination's point of view. And secondly, there are so many factors involved in this topic, so we think that learning the name of these factors is not our cup of tea. But what we don't get to understand is, once we get the concept right, the factors are not going to bother us much. So in this 1 hour video, I'll be covering all the concepts of cell cycle regulation in just one go with the help of some flowcharts and graph. And if you will watch this video cautiously till the end, the concept will be at your tips and you will get to learn the names of the factors very easily. And in that way, you can solve all the questions from this topic in just one go, including the 4 marks question. Just a quick disclaimer before we begin. I know it's quite tiring and boring to watch any video continuously for 1 hour. So it's up to you if you want to take any breaks in between. And for your convenience, I'll be giving the timestamps of different topics of this video in the descriptions below. And you can jump to any of the topics directly if you want to. And the second thing, this is not a sponsored video. You can learn this topic from any of the standard books of cell biology. But for summarizing all the facts and figures for this video, I have referred the Fundamentals of Life Sciences Volume 2 by Catalyst Publications. So if you want to grab that book, the link is in the description. So without any further ado, let's get right into the topic. First of all, let's start with what is cell cycle. Well, we already know about that, right? Cell cycle is a series of events in which a cell grows, replicates its DNA, and then divides mitotically into 2 daughter cells. And then this series of events repeat in the daughter cells as well. So it is a cyclic event. Basically, we divide the cell cycle into 2 major parts, the M phase and the interphase. M phase is the phase where the cell divides mitotically, and the interphase where all the other metabolic activities of the cell cycle take place. The interphase itself can be distinguished into 3 distinct phases: G1, S and G2. G1 phase is the phase that comes after the mitotic division and before the synthesis of DNA, or the DNA replication phase. And in this phase, the cell prepares itself for the process of DNA replication, and it is where the cell decides if it is going to divide or not. So if the cell decides that if it is going to divide in response to the growth factors and the nutrients available, then the cell moves to the S phase. Otherwise, it stays in the G1 phase, or in other ways, it comes out of the G1 phase into another phase, that is the G0 phase, which is considered out of the cell cycle. After coming to the S phase, the cell starts replicating, and the DNA and the amount of DNA in the cell doubles. And after the S phase comes the G2 phase, where the cell takes a break and analyze if everything in the S phase has occurred properly. So if any damage or mistake has occurred in S phase, first of all the cell repairs itself, and then once everything seems to be fine, everything seems to be fixed, then only the cell moves from the G2 phase to the M phase, where the cell divides mitotically. From the examination point of view, it is very important to know the amount of DNA at different phases of cell cycle, and it can be understood with the help of this graph. But before coming to this graph, we have to understand what is C value. C value is defined as the amount of DNA in an unreplicated gamete of an organism. If I take the example of any diploid organism, for example human beings, right? So our cells will be carrying the DNA which will be equal to 2n, but our gametes, the unreplicated gametes, will be carrying the DNA equal to n. So the C value, which is the amount of DNA in the unreplicated gamete, will be equal to n in the case of diploid organism. But if we take example of any tetraploid organism, for example, so the amount of DNA in the body cell will be 4n, but the amount of DNA in the gamete will be 2n. So the C value in that case will be equal to 2n. So coming to this graph, in the G1 phase, the cell is carrying the DNA which is equal to 2C, that is the normal DNA amount in the cell, and as the DNA replicates in the S phase, the amount of DNA just doubles into the G2 phase, and it becomes 4C. And when we move further, in the M phase, where the cell divides into two daughter cells, in the G1 phase of the daughter cell, the amount of DNA is equal to 2C again, right? So it should be kept in mind. Another important topic from examination's point of view is how the different phases of the cells are arranged, and there are various questions asked from this simple concept only, and it is asked very frequently as well. So let's have a look at that as well. The arrangement of different phases of cell cycle and the duration of each phase can be determined with the help of a very simple experiment. But before we move on to the experiment, let's have a look at the bar I've drawn at the bottom, right? So I have mentioned G1, S, G2 and M phases of the cell cycle at the bar. So this bar represents the entire cycle, but for this instance, let's assume that we don't know what are the different phases of the cycle and we don't have any idea about the duration of each phase. What we all know about the cycle is that entire duration of the cycle, that in a particular time that cell cycle completes, right? So what we are going to do now is we'll take a culture of growing cell and we'll treat it with tritium thymidine for approximately 30 minutes. What tritium thymidine is going to do is it will get incorporated into the DNA of newly synthesized DNA of the cell. That means only those cells will get the tritium thymidine which are in the S phase during the treatment of during this treatment in the cell culture. The cells in the G2 and M phases will not get the tritium thymidine, so they won't get labeled, right? So the cells in the S phase are going to get labeled with the tritium thymidine, and the cell in the G2 phase and M phase during the period will treated it the culture with tritium thymidine are not gonna labeled. Okay, so after 30 minutes we take the sample of cells from the culture, we fix them, and we try to analyze and see the radioactivity in the mitotic chromosomes of the cell. Why mitotic chromosomes? Basically, when we look at the different phases of the interphase, the G1 phase, S phase and the G2 phase, there is not much difference in the chromosome appearance in these phases. But the chromosome in the mitotic phase is quite different, as it is very hardly compressed and compacted. The compaction is up to 10,000 folds, right? So it can be easily distinguished from the other ones. So what we are going to do is we'll see the radioactivity in the mitotic chromosomes only. Okay, so after 30 minutes of treatment of tritium thymidine, what we are going to do is we'll take the sample, fix them and see the radioactivity, and the results we'll get is there will be no radioactivity in any of the mitotic cells. So for this instance, let's assume we have treated it started the treatment at this point, and let's move it to some point here. Let's move it to 1 or 2 hours before we take the sample. So we have treated the culture with 1 or 2 hours with tritium thymidine for 1 or 2 hours before we taking the sample, and still no radioactivity seen in the mitotic chromosomes. And what does this indicate? That there is some time interval and gap between the S phase and the M phase, right? Because if these two phases were in continuation, we would have seen some radioactivity, but there is a time interval, and what we named that interval is the G2 phase. Now, to know the duration of the G2 phase, what we are gonna do is we'll simply try treating the culture with tritium thymidine much more earlier, and the first cells which are gonna get labeled is in the last stage of S phase, right? So the cells which are in the last stage of the S phase will be get labeled first, and in this way we'll be getting the duration of the G2 phase totally, right? To know the duration of S phase and M phase, well, it can be calculated directly. Let's assume the S phase first of all. So it can be calculated with a very basic, keeping a very basic principle in mind, that at any instance in a cell culture, the percentage of cells in any specific phase is going to represent the percentage of time of that phase into the cell cycle, right? For example, if we see 50% of the cells in a culture in S phase, right, and the entire length of the cell cycle is suppose 10 hours, so we can assume that 50% of the 10 hour, that is 5 hours, is the duration of S phase. Okay, and in the same way we can calculate the duration of M phase as well. So when we add up the S phase, G2 phase and M phase and compare it with the entire cell cycle length, we see that there is still an interval which is unknown to us, and we named that interval the G1 phase. So there are many questions asked from this simple concept, and what they generally ask is the questions from this graph, and now we can interpret this graph very easily. On the y-axis is the percentage of labeled mitotic chromosomes, and on the x-axis is the hours after tritium thymidine addition. So you can see that at the starting there is no labeled mitotic chromosomes found, and this indicates that it is the G2 phase actually, where the cell have already gone through the phase of DNA replication, and none of the newly synthesized DNA got the tritium thymidine, right? But as soon as we reach this point, the last stage of S phase, we get the labeled chromosomes, and so this phase will be this ascending phase of the curve will be representing the S phase, or the last stage of S phase. So they can ask various type of questions from this graph, and this phase is going to represent the S phase, this phase is going to represent the G2 and M phase. So they can put any point on the graph and ask what does that point represent. They can ask for the duration of the cycle, they can ask for the duration of any phase, they can ask according to their convenience. The duration of cell cycle is highly variable, and the most variable phase of the cell cycle is the G1 phase, because there are few cells which divides very rapidly and there are a few cells which don't divide at all. So the cells which don't divide or which divides very slowly, it is checked in G1 phase until and unless it decides to enter the cell cycle. And once the cell decides that it is going to enter into the cell cycle, the cell moves from G1 to S, and once the cell moves from G1 to S, it is bound to complete the entire cycle and it is forced to divide. So what factors keep the cell checked into the G1 phase, and what factors force it to move forward into the cell cycle? This can be understood with a very simple cell fusion experiment. This was done in 1970, and let's have a look at what we did in the cell fusion experiment. So we tried to fuse two cells in different phases of the cell cycle and see the result on the chromosomes of these two cells. Firstly, we fused the cell in the G1 phase with the cells in the S phase, and what result we get? The G1 nucleus show replication. That means there are a few cytoplasmic factor in the S phase cell which force the nucleus which force the DNA in the G1 phase to start replication, and as soon as we fuse the cells, the G1 DNA starts replicating. In the second fusion, we fused S phase cells with the G2 phase cells, and we saw that the G2 phase cell, as it has already gone through a cycle of DNA replication, it does not show any replication again when it was fused with the S phase cell. So that means there are a few factors into the G2 cells that keeps the replication checked, even if there are some activators of replication from the S phase cell cytoplasm. When we fuse G1 cell with the G2 cell, we saw that the G1 cell entered the S phase according to its timeline, and there are no other changes. When any of the cells are fused with mitotic cell, then there is seen a compaction in the chromosome. Even if it was G2 phase cell or S phase cell, the chromosome in these cells got compacted, and when it is fused with S phase cell, the mitotic cell fused with S phase cell, we saw that the S phase chromosomes compacted, and as a result we got the DNA fragmented. So this is how it worked, and again a very crucial topic from the examination's point of view. As I said earlier, the G1 to S transition in the cell cycle is very crucial, because it is the G1 phase where cell decides if it is going to enter the cell cycle or not. So if the cell loses control over this point, a cancer-like situation may arise, right? And the cell cannot afford to have that. The second crucial point in the cell cycle is in the G2 phase, where the repair mechanism takes place. See, the G2 phase comes after the S phase, where DNA replication is taking place, and in the newly synthesized DNA, if there will be some errors, if there will be some mistakes, this DNA is going to be passed to the daughter cells. So these errors will be passed to the daughter cells as well, and the cell cannot afford to have that. The third crucial point is in the mitotic phase, or precisely to the metaphase and anaphase transition. Why it is crucial? Because in metaphase, the chromosomes are aligned in the center of the cells, and during anaphase, the sister chromatids separate and moves towards the opposite pole. So during this movement, there are huge chances that damage to the DNA can happen, and there can be breakage in the chromosomes. So again, this is going to be passed to the daughter cells, which the cell cannot afford. So to regulate these very crucial points and other factors, there are some regulatory mechanisms in the cell cycle, and central to the idea of regulation in cell cycle, there are two very important characters to the story. First one is the cyclin, and second one is the cyclin dependent kinases. What are these characters? Let's have a look at that as well. So what are cyclins and CDK? Well, it is a heterodimeric protein in which the CDK works as a catalytic subunit and the cyclins work as a regulatory subunit. Talking about the CDKs, these are 30 to 40 kilodaltons in weight, and it is a serine threonine kinase. That means it phosphorylates the serine and threonine residues of the target protein, but it cannot work upon its own. By its own, it is inactivated. How? If you look at the 3D structure of a CDK, you will see that the active site of CDK is covered with the T-loop protein, and it opens up, or it is visible only when it is bound to its respective cyclin molecule. So when a cyclin molecule come and bind to the CDK, the active site is visible now for the target protein, and that's how it is activated. But this activation is only partial. For the complete activation of the CDK molecule, another kinase protein, which is known as the CDK activating kinase or CAK, binds to and phosphorylate the threonine molecule near the active site of CDK to make it completely active. As you can see in this table, there are various CDKs which binds to the respective cyclins. The binding of cyclin and CDK is very specific. It's not like that any cyclin can bind to any of the CDKs. So in case of vertebrates there are various CDKs, but in case of yeast there is only one specific CDK which binds to its respective cyclins according to which phase it is going to work upon. So coming to the cyclins, it is named cyclin because its level fluctuates throughout the cell cycle, and it binds to CDK and activates its kinase activity. Thus cyclin, what it does is, cyclin triggers when combined with CDKs, the complex triggers the phase transition and moves the cell cycle into the forward direction and prepares the cell for the next phase, right? So in this way the cell cycle moves ahead. Now there are various cyclins. First one is the G1 cyclin that works in the G1 phase. It is quite different from the other three cyclins as in two aspects. First one being that it does not trigger any phase transition, and second one being that it communicates with the extra outer environment of the cell, through which it senses the external growth factors and inhibitory factors and let the cell know if it is ready to divide or not. The second cyclin, the G1S cyclin, in the case of vertebrate it is cyclin E. It binds to the CDK2 and it helps the transition from G1 to S phase. The S phase cyclin, which is the A cyclin in case of vertebrates, binds to the CDK1 or 2, and it helps in again the phase transition and also the replication of DNA. And finally the M cyclin, which is cyclin B in case of vertebrates, and binds to CDK1, and what it do is it helps in transition from G2 to M phase and also the progression of mitotic phase. How the level of different cyclins changes throughout the cell cycle can be interpreted from this graph very easily. So let's take all the cyclins one by one. Firstly, let's look at the G1 phase cyclin. The level of G1 phase cyclin remains almost constant throughout the cell cycle, so it's not mentioned in this graph. Talking about the G1 S phase cell cycle, its levels starts increasing from the basal level into the last stage of G1 phase, and it helps in the transition of G1 to S, and ultimately in S phase, in the beginning of S phase, when the cell entered the G1 S phase, its levels goes down again and kept lower throughout the cell cycle. The S phase cyclin, its level started increasing again in the last stage of the G1 phase, and it helps in transition of G1 to S and also helps in the DNA replication, and its levels continues to be high until and unless the metaphase to anaphase transition. After that its level goes down to the normal. The M phase cyclins, its level changes at the G2 in the G2 phases, and it starts growing up, and it helps in the transition from G2 to M, and then suddenly after metaphase to anaphase transition its levels goes down because of some APC proteins. We'll be seeing that later. So in this way the level of cyclins changes throughout the cell cycle. Before we proceed to the stepwise procedure of cell cycle regulation, I would like to introduce a few more factors in front of you that we are going to see throughout the regulation cycle, right? So, and if you understand those factors here, if you understand the functionality of these factors here, it will be easier for you to analyze and understand the stepwise procedure when I draw the flowchart. So let's have a look at that as well. Now, as you all know by now that the CDK activity depends a lot upon the availability of cyclins, but cyclins are not the only factors that control the activity of CDK. There are a lot of other factors, the cell cycle inhibitors and cell proliferative factors, that control the activity of CDKs. So what are these? Let's have a look at those one by one. So at the S phase, CDK activity is checked, and the CDK is inhibited by the CKI protein. What is CKI? The CDK inhibitory proteins, right? What does that do? Well, they come and bind to the cyclin CDK complex and alter the active site of the CDK. So in this way the activity of CDK is checked. And what are these proteins? It is SIC1 in case of yeast and P27 in case of vertebrates. But this type of inhibition into CDK is reversible, and it is removed. This type of inhibition is removed by a protein known as SCF. What is SCF? It is SKP1 Q-lin F-box protein complex. What does it do? It degrades the SIC1 and P27 proteins. How? You can see here that till the end of G1 phase, right, the levels of SCF are low, but at the G1S transition it levels starts to increase, and it kept high throughout the cell cycle until the metaphase to anaphase transition. So until and unless no cyclin CDK is required, it was the levels of SCF was low, and once it was required, the SCF levels increased and the cyclin and CDK levels are higher now. Now the second type of inhibition we are going to see is at G2 phase, and this type of inhibition is inhibition by another kinase protein, that is V1 kinase. What does it do? It comes and phosphorylates the tyrosine at 15th position and threonine at 14th position of the CDK, and in this way it alters the active site of the CDK protein. So again, this is a reversible process, and once it is inhibited, it can get activated again with the help of a phosphatase protein, the CDC25 phosphatase. What does the CDC25 phosphatase will do? It will remove the additional phosphate groups added by the V1 kinase, and in this way the activity of CDK will be regained. So the third way with which we can inhibit the activity of CDK is by the destruction of cyclins itself. And you know all that once the cyclins are degraded, no cyclins will be available and the activity of CDK will be lower. And how it is done? It is done with the help of ubiquitin proteasome pathway. Now this is very crucial point in the cell cycle, because the destruction of cyclins with the ubiquitin proteasome pathway ensures only the forward movement of cell cycle is possible, because once the cyclins are destructed with proteolytically, there will be no cyclins available again, so the cells will be forced to move in the forward direction only. And how the ubiquitin proteasome pathway works? Well, it works with the help of a protein that is known as APC, which is anaphase promoting factor, also known as cyclosome. The anaphase promoting factor binds to two factors and perform two different tasks. Let's have a look at this with the help of this graph. So firstly, the anaphase promoting factor APC binds to CDC20. Now till the G2 phase, till the end of G2 phase, its level are low, but at the G2 to M transition the levels start increasing, and it is at peak during the metaphase to anaphase transition. And during the metaphase to anaphase transition, it helps in metaphase to anaphase transition. How? The sister chromatids at the metaphase are held together with the help of a protein known as securin. So what the securin do is, it does not allow the detachment of sister chromatids, right? So these proteins, the APC CDC20, they will come and destroy the securin protein, and not only it causes the destruction of securin, it also activates another protein that helps in the separation of the sister chromatid, and that protein is known as separase. So in this way they destroy the securin and activate the separase protein, and in this way they cause the metaphase to anaphase transition. And once the metaphase to anaphase transition have occurred, its levels start declining. How? Because this APC CDC20 complex is now hydrolyzed with the help of, it is now hydrolyzed by another protein known as CDH1. And what does that CDH do? It replaces the CDC20 and binds to the APC CDC20, and so its level are low till the metaphase to anaphase transition, and from the metaphase to anaphase transition its level starts increasing, and it is at peak towards the end of mitotic phase. and this is responsible for the destruction of mitotic cyclins, right? So, and this is very crucial for the cell to exit from the mitotic phase, and the levels of APC-CDH1 are kept high till the end of G1 phase, and in the G1 phase it ensures that there is no premature accumulation of the S-phase cyclin and CDK. So this is how the different factors are kept checked by the help of APC protein. We'll be seeing the details later. And the way by which the CDK activity is checked is by the movement of cell cycle regulators in different compartments of the cell. Now what does that mean? It can be understood with the example of cyclin B1. If you remember the table that I drew here, the cyclin B is the mitotic cyclin, right? So until the end of G2 phase, the mitotic cyclin shuttles between the nucleus and the cytoplasm, and towards the end of G2 phase it starts accumulating in the nucleus, and only once it is accumulated in the nucleus, then only it can cause the activation of CDK, and then only the G2 to M phase transition can take place. So this is how these factors work. Keep it in mind, the functionality of these factors are going to play a huge role in the regulation part. So now let's have a look at different phases of cell cycle regulation, and starts with how cell enters the S phase. Okay, so now when we have got the idea of all the cyclins and CDK and other factors responsible for the cell cycle regulation, we can now easily look into each phase and how the transitions take place. And we'll start with the yeast cell, then we'll be seeing in the metazoans as well, and firstly we'll be looking into the entry into S phase. So for entry into S phase, we all know that G1 S cyclins and CDK and S phase cyclins and CDK are required, and for the synthesis of G1 S cyclins, SBF transcription factor is required. But in the G1 phase, SBF transcription factor is bound to the Y5 transcription repressor, and in this state SBF transcription factor cannot work and G1 S cyclins cannot be synthesized. So how this all happens? Well, when there are high nutrients level and enough growth factors, CLN3 protein, which is CLN3 gene, which is the gene for G1 cyclin, if you remember from the table, starts synthesizing the CLN3, which is the G1 cyclin. And as soon as the CLN3 is formed, it forms complex with the CDK. Remember, in case of yeast, there was only one CDK, which was CDK1. So when the CLN3 CDK complex was formed, it starts phosphorylating the Y5 protein into the nucleus, and as the Y5 protein gets phosphorylated, it comes out of the nucleus, and the SBF is now free to induce the transcription of G1S cyclins. And when the G1S cyclin transcription start, the CLN1-2 cyclins are formed, and then it forms complex with the CDK, right? So once the G1S cyclin complex is formed, now the cell is ready to go into the S phase. But there is a problem. Remember when I said that CDK is also inhibited by some CKI inhibitors. So in this case, in case of yeast, which is SIK1. Now we have to remove this inhibition in order to make the transition from G1 to S. So how do we do that? Well, it is quite simple. SIK1, when gets phosphorylated at least 6 sites by G1S CDK. One thing I want to tell you is SIK1 only inhibits the S phase and M phase cyclins and CDKs. It does not inhibit the G1 and G1S cyclins and CDKs. So in this way, when enough of the G1S cyclins and CDK will be formed, when the G1S cyclin CDK will be at peak, there will be enough concentration of G1S cyclin and CDK, then only the phosphorylation at least 6 sites will be happening to Cyc1, and as soon as the Cyc1 gets phosphorylated at its 6 site, it is bound to get degraded by the SCF protein. SCF, as I told you earlier, right? So it is a switch like response. So when the phosphorylation takes place, it suddenly the level of Cyc1 goes down, and suddenly the level of S phase CDK rises up. So it is a switch like response from G1 to S phase transition. One thing just I left here is, when the G1S cyclin, which is the CLN1 and 2 in case of yeast, is formed, it works as a feedback loop, and it also started phosphorylating the Y5 protein, and as a result more Y5 are phosphorylated, and as a result Y5 move out of the nucleus more quickly, and the transcription of the G1S cyclin induce more rapidly. So after we saw the entry of cell into the S phase in case of yeast, now let's see the entry of cell into the S phase in case of mammals. Almost all the steps are similar, but what's different is the name of cyclins, the repressors, the transcription factors, and also the CKI, which is P27 in case of mammals. So E2F transcription factor is required for the synthesis of G1S and S phase cyclins, and it is bound to retinoblastoma, which is repressor of E2F transcription factor, and in this bounded state it cannot synthesize the G1S and S phase cyclins. So what happens is, G1 cyclins, as we know that its level is constant throughout the cell cycle, but when the growth factors are enough and the cell decides to come into the cell cycle, the level of G1 cyclins start increasing gradually, and then it binds to the G1, then it forms a complex with a known as G1 cyclin CDK complex, right? So what this complex do is, it starts phosphorylating the retinoblastoma, and this retinoblastoma phosphorylation is kept on throughout the G1 as G2, G2 and M phase, okay, with the different cyclins. And it is necessary because this retinoblastoma protein you will be seeing in the many developmental pathways, and it is very important protein in case of cancer as well. You will be seeing that in case of cancer, retinoblastoma, mutation in retinoblastoma can lead to various causes of cancer, because once it is mutated, it won't be able to disrupt the E2F transcription factor, and E2F will continuously synthesizing the G1S and S phase cyclins, so the cell cycle will continuously move on. So let's see what happens in this case. Once the phosphorylation of retinoblastoma takes place, it moves out of the nucleus, and now the E2F is free to synthesize the G1S and S phase cyclins, and as the S phase cyclin is synthesized, cell is ready to move to the S phase, but the problem is still here, that it is inhibited by the CKI, which is P27 in case of mammals. So how the P27 is degraded, and what's the difference between the yeast and the mammalian cell? Let's have a look. P27 in the case of mammals, the biggest difference is that it inhibits the G1S CDK as well. Remember, in case of yeast I said that Cyc1 only inhibits the S phase and M phase CDKs. It does not inhibit the G1S CDK. So this is a big difference, that P27 is inhibiting the G1S CDK as well. Now it is degraded by two pathways. First, the MAP kinase pathway. What is MAP kinase pathway? I am not going to go into the details because it is the part of cell signaling, but what happens is, through this pathway the P27 gets phosphorylated and it comes out of the nucleus, and once it comes out of the nucleus, it is going to get degraded with by the ubiquitin proteasome pathway when it interacts with the KPC ubiquitin ligase. The second method by which the degradation of P27 takes place is at G1S transition, when the high levels of G1S CDKs will phosphorylate the P27, and then it is going to degrade it, as in the case of yeast, by the SCF protein. One more important function of G1S CDK is that it dissociates the APCC APC-CDH1 complex. Remember, APC-CDH1 complex is necessary for the cell to move out of the mitotic phase of the cycle, and the APC-CDH level is continue to be high in the G1 phase. But once the G1 CDK is high, once the cell decide to go into the S phase, the G1 CDK will phosphorylate the CDH1, and in the phosphorylated state it will get dissociated from the APC, and now the S phase cyclins can get accumulated. This is a reversible process, and these two can associate together only when the mitotic CDK decline after the metaphase to anaphase transition. And this reversal can take place with the help of a phosphatase known as the CDC14 phosphatase, which will remove this phosphorylation from the CDH1, and now it can associate to the APC. Just keep in mind that CDC14 phosphatase work only in case of yeast, not in case of mammals. There is a phosphatase that works in the case of mammals, but the name of that phosphatase is not known yet. So the S phase cyclins is now free, and it will help in initiation of replication as well. And there are various proteins to the initiation of replication. I am not going into the details because it's not relevant to the topic I am discussing here. The ORC, CDC6, CDC1 are the different MCM helicases are going to get loaded into the DNA. But what's relevant from this to this topic is why the DNA replication takes place only in the S phase of the cell cycle. And the biggest reason to that is that MCM helicase, which is responsible for the unfolding of DNA at the site of origin, can be loaded to the DNA only when the CDK and cyclin level, all the CDK and cyclin level are low. So it can be loaded to the DNA only in the G1 phase. But of course it can be activated only once the cell enters the S phase. So once it gets activated in the S phase, it will get phosphorylated and it will dissociate. So this is how the origin of DNA replication is going to take place, and this is why the DNA replication takes place only once in the S phase. And because after the S phase, any cyclin and CDK level is not going to get down, so MCM helicase cannot load it to the DNA again. And after the DNA replication, there will be formation of sister chromatids, and these sister chromatids will be joined together with the help of cohesin protein. Remember the name cohesin proteins and its 4 subunits: SMC1, SMC3, SCC1 and SCC3. These are crucial in the case of checkpoints. Right? So till now we have discussed how the cell enters into the S phase, how the DNA replication takes place, and after the replication again there is a part of repair or mechanism which takes place in the G2 phase, which is again irrelevant to the topic. So what we are going to do now is we are going to see how the G2 to M transition takes place and how a cell enters into the mitotic phase of the cell cycle. So let's have a look at that. For moving into the mitotic phase, we will require the mitotic cyclins and CDKs. So let's see how these mitotic cyclins and CDKs are synthesized and how the complex forms. We'll start with the mitotic cyclins first. And remember when I said that APC-CDH1 is the protein in the G1 phase, which is inhibiting the premature accumulation of the S phase and M phase cyclins. So once it was degraded or dissociated with the help of G1S CDKs, now there are no factors to inhibit the synthesis of cyclins of mitotic phase. So the mitotic cyclins start synthesizing in the S phase. But the problem is with the mitotic CDKs, which is still inhibited by the V1 kinase. And to remove this inhibition, what will be required? Will be requiring the CDC25 phosphatase. So the rapid activation of CDC25 takes place as a kind of feedback loop with the help of this mitotic CDK. When the CDK levels are high, what will it do? It will this CDK will phosphorylate the CDC25 phosphatase, and as a result of this phosphorylation, the CDC25 phosphatase will be active, and now it can remove the phosphate group added by the V1 kinase. So it's a kind of feedback, right? Don't get confused with what's phosphorylation and dephosphorylation. Just remember one basic concept, that whatever is kinase is adding the phosphate group, and whatever is the phosphatase is removing the phosphate group. So CDK being a kinase is adding the phosphate group to the CDC25 phosphatase, and CDC25 phosphatase being a phosphatase will remove the phosphate group added by the V1 kinase. Now CDC25 have a variety of roles, like there are few more proteins to this family, like CDC25A, which removes the inhibition from the G1 as CDKs, and CDC25C, which removes the inhibition from by the V1 kinase. So now once the inhibition by V1 kinase is removed, now the mitotic cyclin and CDK can form the complex, and once the complex is formed, now the cell is ready to enter the mitotic phase, right? But the role of mitotic cyclins and CDK are not over yet, because these mitotic cyclin and CDK complexes are about to play very vital roles in the process of mitosis as well. How? Let's see. Firstly, initially they are attached to the centrosomes, and while attaching to the centrosome, what did they cause? They cause the centrosome maturation, which is required for the mitotic division, right? And once the centrosome maturation have taken place, the mitotic cyclin and CDK complex move to the nucleus, where another very vital process is taking place, which is the chromosome condensation, and they assist in the chromosome condensation as well as in the nuclear envelope breakdown. So how the chromosome condensation takes place, and first of all, why chromosome condensation is important. See, for the chromosome to be segregated to the opposite poles, only the factors for the segregation are not enough, because it is necessary for the chromosomes to be packed in a compact structure so that it can travel without any damage to the opposite poles, and for that purpose the chromosome needs to be condensed, and this condensation is led with the help of mitotic cyclins and CDK. What do the mitotic cyclin and CDK do? They form a collaboration with the two other proteins, the polokinase and the aurora kinase. Remember, in the case of S phase, where the S phase cyclin was collaborating with the replication factors to assist in the process of replication. Almost similar mechanism is here as well, where the mitotic cyclin and CDK are collaborating with the proteins of mitosis to assist in the process of mitosis. So what does the polokinase and aurora kinase do? Well, they have their own roles as well. Polokinase plays a vital role in the spindle fiber formation, and also it plays some role in the chromosome segregation as well, right? And talking about the aurora kinase, this also plays some role in the spindle formation, and along with that it checks if the chromosomes are attached to the spindle fibers correctly or not, right? So in this way the activity is ensured by these kinases, right? So the collaboration of CDK with these kinases also lead to the formation of compaction of DNA. How? The compaction of chromosomes by condensing takes place only when the CDK collaborate with aurora B. And what does that collaboration ensure? Once the mitotic CDK collaborate with the aurora B kinase, it phosphorylate a subunit of histone protein, that is H2A, and only when the H2A is phosphorylated, the condensin can attach to the chromosome. First of all, what is condensin and how it is attached to the chromosomes? Well, there are many aspects to the chromosome compaction, like untangling of the intervened DNA by the topoisomerase 2 and the consecutive loop formation, right? So in the, what does the condensin do? It attaches the consecutive loops together with the help of interchromosomal linkage, right? So this is the function of condensin, that it helps in the compaction of chromosome. And how does it attach to the chromosome? Well, as I said earlier, once the H2A subunit of the histone is phosphorylated, there is a way for the condensin to attach to the chromosome now, and it is attached to the chromosome. So in this way it ensures the compaction of chromosome, but compaction of chromosome is further required, and that happens with the dissociation of cohesins cause further compaction, right? And how does the dissociation of cohesin takes place? Remember, cohesin, I told you earlier that it ensures that the sister chromatids are attached together until and unless they are required to move apart in the metaphase to anaphase transition. So a part of cohesins is removed by phosphorylation by polo and aurora kinase, right? So once the phosphorylation takes place, major part of the cohesins are removed, but yet the part that is attached to the centromere, the part of cohesin that is attached to the centromere, is still not removed, and that part is crucial for the chromosome segregation, so it will be removed later. And once the cohesins are removed, now the entire chromosome is free for more condensation, and the condensins work on that part of chromosome as well, and it is compacted up to 10,000 folds. So now our chromosome is compact and ready to move apart to the opposite poles. So in the next step we'll be going to see how the chromosomes segregate and move to the opposite poles. The prerequisites for chromosome segregation is the criteria that if chromosome is properly condensed and if chromosomes are properly attached to the spindles. These criteria are checked by the proteins of some checkpoint pathway. The protein of checkpoint pathway inhibits the APC-CDC20 until and unless every kinetochore is attached to the microtubules and tension is applied to all the kinetochores. And if everything is fine, every tension is okay, then the tension in spindles is sensed by the checkpoint proteins, and now the APC-CDC20 is activated. And you all know, when APC-CDC20 is activated, it is going to degrade the securin with the help of ubiquitin mediated ubiquitin proteasome pathway, right? And the separase is going to get activated. Separase is basically inhibited by the mitotic CDK, but mitotic CDK is going to be inhibited by the APC-CDC20. So once APC-CDC20 inhibits the mitotic CDK, the separase is going to get activated, and now what separase do? It will cleave the RAD21 subunit of the cohesin. Remember the subunits of the cohesin I told you. The RAD21 is also known as the SMC1 subunit of the cohesin. So now the cohesin is removed, and all the studies done so far proves that the anaphase movement is possible only if the cohesins are removed. So once the cohesins are lost, the anaphase movement is triggered and the chromosomes move apart. Now once the chromosomes have moved apart, the cell would now want to exit the mitotic phase and enter the new cell cycle. So how that happens? At the anaphase, what happens is CDC14 phosphatase is activated by the GTPase signaling. And what the CDC14 phosphatase do? This will now remove the phosphate group from the CDH1, right? And this CDH1 will now hydrolyze the CDC20 and bind to the APC. So the APC CDH1 complex will be formed, and this APC CDH1 complex, it will do? It will degrade the mitotic cyclin, and that's how the cell will exit the mitotic phase. Just one thing, that the SIC1 dephosphorylation is also taking place here. So now the cell is ready to move into the G1 phase, and the S phase proteins will be inhibited by this CKI protein. So with this we have came to an end to this marathon episode of cell cycle regulation, and I hope that this video will help you to build the concept of this topic, and I really appreciate if you have came this far along with me. But before we wrap up the things, I would like to give you a quick recap of whatever we have learned so far, and then I'm going to discuss a very small but very important topic from examinations point of view. So what we have learned so far? Well, firstly we started with what is cell cycle, and we saw the different phases of cell cycle. Then we came to a few experiments through which we saw what are the arrangement of different phases of cell cycle and what are the durations of the different phases. Then we saw the cell fusion experiments, with which we came to know that there are some cytoplasmic factors responsible for transition from one phase to other. Then we learned about the different cyclins and CDKs and the other factors responsible for the regulation of cell cycle. And then we moved ahead and saw the different phases and how the transition actually takes place. Now I want to discuss a checkpoint pathway or a DNA damage response, that how a cell responds when a DNA damage takes place. So central to this pathway are two proteins known as ataxia telangiectasia mutated, that is ATM, and ataxia telangiectasia RAD3 related protein, that is ATR. Well, what does these two proteins do? They basically sense any kind of DNA damage. ATM being a very specialized kind of protein, it senses only the double-stranded damages, and ATR senses any kind of damage in the cell. So when these cells sense any kind of damage, they are recruited to the site of the damage, and then they recruit some other adapter proteins to that site, which is CHK2 and CHK1. Well, these proteins, now what will they do? They will either repair the cell, they will either repair the cell, or they will arrest the cell into that phase, or they will lead the cell to the apoptotic pathway. So if possible, they will activate the repair pathway. I'm not going into the details of that, or they will inhibit the activity of CDC25. And what was CDC25 doing? It was activating the CDKs. So once the CDC25 is inhibited by these adapter proteins, the CDK cannot be activated and the cell cycle will be arrested. Now the third way they can work is they will be activating the transcription factor P53. What this transcription factor will do, it is responsible for the production of an inhibitor, that is P21. So once the transcription factor is activated, an inhibitor will be produced and the cell cycle will be arrested at that phase. Or the other thing that this P53 can do is this can activate the apoptotic pathway. So in this way the cell responds to the damaged in DNA. Well, there are several other checkpoint pathways as well, but those are just worth reading. So that's it for this video. Thank you so much for watching this long. Let me know if you liked my work, share this video to spread my words, and I would really appreciate if you take a moment to subscribe to this channel. Comment section is open for every query and suggestion, and suggest me what should be the topic of my next video. Catch you guys in the next one. Bye bye.